let's do get underway. Really, again, appreciate this morning's conversation and the presentations helping us to, well, wondering if I should just, helping us to define what is and is not synthetic biology and to understand a little bit about, a little more, in fact, about some of the science involved. This particular session, we are going to focus, we have asked our experts to focus more on applications. We'll try to use a similar format, have our speakers present for a brief period. You've got the timer in front of you. Make sure after all of you have presented that the Commission has time to ask questions, and then we'll try to throw it open. Not try to. We will certainly throw it open to the public for other questions they may have. Our first speaker we've heard all about, Dr. Craig Venter, is the founder and chairman and president of J. Craig Venter Institute. Founder and CEO of Synthetic Genomics Incorporated. We have been talking all morning about some of the many feats that his team has accomplished, including the most recent demonstration that a cell of one microbe species can incorporate the genome synthetically assembled from another cell. He is a member of the National Academy of Sciences, the recipient of many awards, including the 2008 National Medal of Science by President Obama. We are delighted to have you here, Dr. Venter, and look forward to your comments.
Thank you very much. It's certainly a pleasure to be here, and I'd like to start off by thanking President Obama for asking the Commission to take on a review of what I think is a very critical topic. In fact, we asked for the first ethical review at your institution, the University of Pennsylvania, in the mid-1990s before we did the first experiment, as soon as we knew we were going in this direction. And the results of that review after 2 years was published in Science in 1999, and I think that was the first, to my knowledge, scientific ethical review before the science was done. So I think we've set a new precedent in this field of actually going against my usual trends of asking permission first, usually asking for forgiveness afterwards for moving things ahead. But that was just the start, and I think this is now the third administration to deal with some way with synthetic genomics. President Clinton actually had a special session in the White House where myself and others addressed the issues of making synthetic viruses, particularly around the sequencing of the smallpox genome, which my team did in the early 1990s. And all these issues about public availability of such critical information came up at that time, and the U.S. government came down on the side of open dissemination of this information. Since then, the Sloan Foundation has funded now a few studies and reports with my institution along with MIT that was published last year. On publication in 2003 of the synthetic Phi-X174 virus, because that work, in fact, in contrast to what you heard, was actually funded by the U.S. government. It was funded by the Department of Energy. After lots of deliberations within the executive branch of the government, they came down on the side again on open publication of this information and the methodology. And it was announced, as all our studies have been done, with a peer-reviewed scientific publication. Most of these have been in a series in the journal Science. And it was announced publicly with a press conference with the Secretary of Energy. So there has been government involvement almost at every level of this from the beginning. And part of the agreement that was had at that time was the formation of the NSABB to look at dual-use research. The National Academy of Sciences, the so-called Fink report, has looked at dual-use research. And just recently, the DOE just gave another grant to Bob Friedman and Michelle Garfinkel at my institution to look further at some of the ethical uses and limitations. But even though we've been discussing this broadly for over a decade, it's been in the world press for well over a decade. I think our recent publication was the first time most people, and probably most of you, actually heard about synthetic genomics. So I think this commission can take it to the next stage, and as Drew Indy said, this technology is moving very rapidly in a progressive fashion. Let me help with some definitions, because I think there was clearly some confusion that came out of the first panel. The fundamental differences between what we do and what's been done before and how we define synthetic genomics is we start with digital information in the computer from all our reading of the genetic code. My team sequenced the first genome of a living organism in 1995, and we went from having the ACs, Gs, and Ts to ones and zeros in the computer. Synthetic genomics, as we've defined it, is starting with those ones and zeros, remaking the software of life, and then activating that in cells. And Dr. Basler was correct about the pioneering work of Kornberg in the 1960s. In fact, President Johnson had a huge announcement on how the first synthetic life had been created at that time. But the difference is Kornberg and team did not know what the sequence of the Phi X genome was. They copied it with DNA polymerase, so it was the equivalent of a Xerox piece of DNA. But the important thing is when they put that DNA into E. coli, it started producing the viral particles. The difference is what we did in 2003 is we started with the digital code of the Phi X genome in the computer, made the genome from the 4 bottles of chemicals that Drew Endy showed you, and activated it, showing that the synthetic molecule could be activated in the same way that normal DNA or even Xerox copies of DNA could be. The big challenge was going the next stage to try and do this with a bacterial cell. And our whole motivation for doing this was trying to understand a minimal cellular life form, trying to get down to minimal gene sets. The technology in molecular biology tool sets does not exist until we did this work to be able to do that. So there's many distinctions, but the single most important one is we start with digital code in the computer. And that was the concern during the Clinton administration about smallpox. If we could regenerate it from the digital code, then destroying the virus had no impact on national security or human health. And that's been an ongoing discussion since. So we scaled up the synthesis abilities over the last decade, first with a 500,000 base pair genome, but we were not able to boot that up in a cell. There were 2 aspects of this work. One is the chemical synthesis of the DNA, and the other is booting it up. DNA is the software of life. The notion that came out of first Kornberg work and then our work with the synthetic Phi X genome is the DNA, the software, builds its own hardware. So it's irrelevant, evoking vitalism arguments that were debunked 80 years ago. There's components in the cell. George Church and others are going to be using those components, and we think we will as well, to have cell-free systems that can be reconstructed. I think it's interesting science, but it is really irrelevant to these arguments of how you boot up a new piece of chemical software. So one of the most important experiments we did was in 2007, where we isolated the DNA, the chromosome, from one species, transplanted it into another cell, replacing the DNA in that cell, and it converted the cell we transplanted the DNA into the species that we isolated the DNA from. So it's like putting different DNA in you and converting you to another species. We can do that at the single cell level. Doing it at the multiple cell level is perhaps decades off, but not centuries away. So these are important concepts about starting with digital code, starting with DNA. It is software, and it does build its own hardware. I think it's an interesting science question. What do we need? Do we need some ribosomes and a few tRNAs and some other things and some lipids? And can we get some cells boot up from that? I think that's going to be important about understanding origins of life. But we are, in fact, building upon 3.5 billion years of evolution. The argument's because we're using genes that we've discovered versus inventing new genes is kind of a spurious one. It's like trying to say Tesla didn't build a new electric car because they bought the batteries from one source and they bought an electric motor from another source. They combined that to make a Tesla, which is a pretty exciting electric car. My team has discovered the majority of genes known to science. We're up over 40 million. There were less than 1 million when we started. These are going to be the future design components. I think BioBricks are an important teaching tool. It's great for getting students involved. But the number of genes on this planet, I'm sure, will top out somewhere over 200 or 300 million. We're dealing with a lot of design components. Nobody's going to patent them. And I think combining those in new ways, now using these tools that we had in the proof of concept experiment, is what the future of this field is going to be. We couldn't do any of this until we did the study that was just published in Science. We were able to make these really large pieces of DNA, but until you can boot them up in a cell and get them activated, it was an interesting academic exercise. So it's very different from what's happened before in molecular biology. This is a new set of tools starting from a new vantage point. And as Drew said, it changes a lot of the rules. Scientists sort of controlled who got what, whether they sent them their cell line or their DNA clone for a gene. Now anybody who has access to the internet, if that information's in the public databases, you can download it and you can make those genes. Now any virus sequence that's in the public databases can be pretty readily remade. Fortunately, not all of them is the DNA infective. Smallpox is one of them. We're just having the DNA from smallpox on its own can't just boot up readily. But these tools are there, and it's a different starting point. All you need is the digital information in a DNA synthesizer. So building the pieces of DNA was an interesting technological challenge. Getting it booted up was straightforward biology and molecular biology. None of it is cloning. So these are totally misuses of terms. Cloning means everything and anything to biologists. It's sort of a collecting term. It's making copies of cells. It's making copies of DNA. It's splicing DNA. So we think it's an irrelevant term for what we do. We use the term synthetic cell because every protein in the cell, all the constructions in the cell are derived from the synthetic DNA. The cell that we used as the recipient cell, all its characteristics are 100% gone after a few replications. So everything in the cell that we have is from that synthetic DNA, and therefore we defined it as a synthetic cell. It's a cell that never existed before. Of course, we used copies of existing genomes. I agree with the very statements. We're very early on in our knowledge of biology, but we definitely have new tools now to get there. We're using some of these tools to make new vaccines. So we have a program that is funded by the NIH to make synthetic components of every flu vaccine that we and others have ever sequenced. And we can recombine these and make a new flu vaccine seed candidate in less than 24 hours. And we're working with Novartis, and it's very possible the flu vaccine you get next year will be from these synthetic DNA, synthetic genomic technologies. It was announced last year that we have a program with ExxonMobil to try and get cells to capture CO2 and make basically a biocrude that can go into refineries. We still have not found any cells that can do this naturally at the levels that are required. So at the very minimum, it's going to need extensive engineering. But I'm absolutely certain, at least by the time we get to version 2.0 of these cells, they will be completely synthetic, as will most things going forward in an industrial environment. Definitions are important. The definitions can be found in our scientific publication. I think this is an area that Drew and Dee's students show we're limited more by our imaginations now than any technological limitations. And I think having an intelligent, ethical, legal framework for this new science to emerge in is absolutely critical. Thank you very much.
Thank you. And we appreciate your views and clarification on the definition, and also impressing upon us again the value of the technology coupling with the science. Dr. George Church is our next presenter. Professor of genetics at Harvard Med. He has pioneered technology innovations in reading and writing DNA, co-initiated the Human Genome Project. He directs personalgenomes.org with the goal of enabling open access integration of full genome sequences, environmental and trait data, a goal of working toward 100,000 individuals. very interesting application. And again, this session being on applications, very eager, Dr. George, to hear what you have to say. Thanks for being here.
Okay. So thank you for the time here. As soon as my slides come up, I'm going to talk almost entirely about applications, and it's going to differ a little bit from previous talks in that not going to talk about introductory or definitions, and in particular about what we can't do or haven't done, but what we are doing. So this is my thank you slide and my conflict of interest slide. Next slide, please. Can I control this? Oh, there we go. Perfect. Yeah. Okay. Oh, sorry. And so as a graduate student, I worked with Greg Sutcliffe, another graduate student, to sequence the first semi-synthetic plasmid, which we did at ridiculously high cost, even though we were students. And this has been used in many recombinant DNA efforts that some of them listed here that were really single gene efforts from Biogen and so on. What's wrong with this picture? This fellow is not using safety goggles. He's not properly grounded for electroporation. But the main thing is we've gone well beyond manual genome engineering that I had in the last slide. We have gone beyond minimal cells to these fast, robust, useful cells. We're focusing on lowering costs. We've talked a lot about scaling up, but not lowering costs. I will focus on that. We look forward to whole genomes, but most of what I'll talk about is doing a little bit less than a whole genome, but on a genome scale. And the question is, why do we do things on a genome scale? And then there's safety and security as a main reason for doing things on a genome scale. And then evolution is a unique capability that we have that most other fields of engineering do not have. And my major take-home from all this is that we are going much faster than it appears, and we should not be reassured that biology is not capable of engineering, and that there's no difference between what we're doing and what I did as a graduate student. Why genome-wide? This is the big question. You can talk about how to do things genome-wide, but we really need to know why. And genetic engineering was one or two genes. Genome engineering, as it's commonly used term, is also a couple of genes. It's just done in the chromosome rather than on plasmid. Big deal. Metabolic engineering, you might do a pathway or a small network. Again, 30 genes or less. But genetic code offers us multivirus resistance, some safety measures, and some use of new amino acids. And this truly is genome-wide. And it's one of the few clearly articulated goals, I think, that is genome-wide. Now, the safety component, I think, is incredibly important. And you can this is not meant to just be analogies, these are just images, but we have interoperable parts. These are all from the cars, but the same thing applies in biological engineering now. Hierarchical design, computer design, cost effectiveness, standards and isolation. It isn't sufficient to have a set of rules and guidelines if there isn't testing, if there isn't surveillance. You can do licensing, as we do with driver's licenses, but you have to do surveillance to make sure people are obeying the laws. And then again, evolution is something that's new. There have been recommendations in 2006, on the next slide, 2007, which I think don't go far enough. We talk about preferred practices. We pragmatically talk about federal grantees and contractors. There's a lot more out there than federal grantees and contractors. The Sloan 2007 went a little bit further than this, but we need to have surveillance and enforcement. And so back to my earlier recommendations on really licensing the entire ecosystem in synthetic biology, I think is important. We need to have surveillance and testing of systems that are proposed to go in. And this is not restricted to bacteria. We have a very active human synthetic biology community and human do-it-yourself community. Some of my undergraduates have gone and sequenced parts of their genomes on their own without FDA approval and without really using any special equipment. And this is a whole other subject we're not going to talk about, but do-it-yourself or do-it-ourself biology and bioweather map and so on. We've studied vaccination. It's another topic for another day. Genome engineering, some success stories are the already mentioned one for artemisinin, but also propanediol from DuPont. This is a $400 million project that was very successful. 90% of the theoretical yield. It only involved 8 foreign genes, plus 13 down and 6 up regulations in the E. coli genome. 27 changes was a lot of work back then. I'm going to talk about hundreds of changes that we've incorporated. These are 2 other companies that I helped start that are not in the future but are already making thousands of liters of production scale fuels, either from biomass or from carbon dioxide and light. This is LS9 and Joule. These are making alkanes, diesel and gasoline. And part of this is the success of comparative genomics. You can look through algae and cyanobacteria for those that make trace amounts, as Craig sort of alluded to, just trace amounts of these alkanes by taking fatty acids, reducing them to aldehydes, and then decarbonylating them. But to find those genes, you can look at the genomes that produced and those that didn't produce these trace amounts, and then you can identify the genes and overproduce them. Rob Carlson alluded to this exponential curve. This is actually quite different from his curves, although they're basically the same. What's different is that around 2004 or 5, there was an increase in the rate of this exponential curve from 1.5 to 10-fold. And more importantly, this is a gap between between our recent huge increase due to second generation or next generation sequencing and synthesis, and we're still stuck in the first generation for gene synthesis in those 4 companies and genome synthesis. We're using first generation sequencing and synthesis for the most part. There are 21 next generation sequencing technologies and 21 companies that go with it, and I advisor for about 16 of them. And similarly, there's next generation synthesis off of chips that we've been doing since around 2004. This has lagged a little bit behind for making genes and genomes, but it's certainly terrific for making very short constructs. Getting those working into cells, it's one thing to make DNA, but getting the work into cells, there are many tools. These are protein-based specificity tools, and more general tools, which are DNA-based, homology-based. They don't require specific proteins to put it in precise locations in the genome to make precise changes. But some of these involve single-stranded DNA number 3 and number 4 in particular. And we've automated this in order to bring down the costs and extend our capabilities industrially. One of these is called, or the general term is multiplex automated genome engineering, or MAGE. And this has one particular implementation shown on this slide, but there are many others. You can see it's a catch-all phrase. This one uses single-stranded oligonucleotides that use computer-aided design to optimize secondary structure, to optimize the position and length. You have to have mismatch repair turned off for some of these, and there's special proteins. But the key point is in a few years, we moved from an efficiency around 10 to the minus 4, 1 in 10,000, to 25% to 100%, and now we can get up to 8 mutations per 2-hour cycle, and we can just continue to cycle 8 changes precisely in the genome wherever you want. And you can make up to 1 billion different changes in a population. I'll show you an example where we did 100,000. This is Harris's prototype, a computer design of the upgrade. This is the actual upgrade. This is applying it where we made 100,000 genomes, not one by one, but in a mixture, and it shows the awesome power of accelerated evolution in the laboratory, where we can make these 100,000 genomes focusing all of the changes in the known pathways, including putting in some genes from other organisms, and in 3 days we could get the highest yields we've ever seen for this hydrocarbon lycopene, which makes tomatoes red. It involved on the order of 24 genes. Another project that we've done, which is less combinatorial, less evolutionary based, where we want to make a precise genome which is resistant to multiple viruses and allows new amino acids to be incorporated efficiently and has safety features. Here we changed all of the codons for all the examples of a codon, TAG, into TAA genome-wide in order to free up that codon and allow us to delete the cellular factor that recognizes it. This can be generalized. There are 64 codons of these triplets, and we've targeted 9 of them. This allows us to do 3 things: new amino acids, safety features, and multivirus resistance, which itself is a safety feature. We have these 9, so we've done 1 of these 9 codons that we're targeting out of 64. We've synthesized all the DNA to do the remaining 8, at least a proof of concept on the essential genes. And another topic that's far beyond what we can talk about today probably is a project where we're making mirror ribosomes, and Craig alluded to this, which is an in vitro system that has interesting commercial applications. The key thing here is just changing these 9 codons would require changing just 2.7% of the genome, not the whole genome. But if we're making them these optimal 90 mers, we basically have completely tiled the genome 2.5-fold over. We essentially have remade the genome, even though we've only changed 2.7% of it. And that lies in the future, and it remains to be seen which is more efficient, doing it all synthesis all at once, where we'll probably have multiple failures, or doing it one at a time. And just as a last slide or two is this issue of safety in terms of isolation. You can have physical isolation or you can have biological isolation. The changing of the genetic code gives you biological isolation, and the genes can neither come in nor go out that are functioning. So that is something that I think critics of genetically modified organisms have wanted for years. Hopefully we can provide it. Another way that a third way that it's isolated, aside from physical and genetic, is this metabolic. Dating back to the early days of recombinant DNA, there was this DAP, or diaminopamalic acid, which was used by deleting the biosynthetic pathway, that you made the bacterium dependent upon that. It's not common in the environment, but it does occur, and that's one of the downsides. Some of these other SAC-B or toxin-antitoxin pairs like CCD and maize are used, but as counter selections, but they are ways of having the cell self-destruct. But they have the problem that they can be lost just before you need them, so that they're not ideal. So we think going forward, we're excited about using the new genetic code to allow us to design multiple essential genes to have multiple dependencies on essential amino acids, which have been used already in Peter Schultz's group, methyltyrosine and aminophenylalanine. So in conclusion, just to remind you, where we think we need genome engineering and synthetic biology is in making biology safer than it already is. And this involves really using some of the lessons of other engineering disciplines, interoperable parts, hierarchical designs, computer-aided designs, cost-effectiveness, standards of isolation, testing, redundant systems like seatbelts and airbags, surveillance, very important, not just surveillance of government grantees, licensing at every scale, every part of the ecosystem, and focusing on this ability to evolve both in the lab and outside the lab. Thank you.
George, thank you for that. Your message is loud and clear that the pace of advancement, technological advancement, is astounding, and some of the near-term applications very exciting, and also clarifies the need. I appreciate your last slide, and and as you used it before, helping clarify for us what some of the challenges, engineering challenges, are going forward. Our final speaker in this panel is Crystal Jones Prather. Dr. Prather is an assistant professor of chemical engineering at MIT. She's worked in industry as well as in academia, has been recognized for her work with numerous awards and investments. She is an ONR, Office of Naval Research Young Investigator, She received Technology Review's TR35 Young Investigator Award. She has also the NSF investing in her through an NSF Career Award. She's an investigator in the Multiple Institutional Synthetic Biology Engineering Research Center, funded by NSF. Welcome. We're pleased to have you here.
Thank you very much. And let me start by thanking the Commission for an opportunity to come and speak to you today. The title of this panel is Applications of Synthetic Biology. And what I'm going to do is to try to give an overview of what recent accomplishments in the field have been. And I've done this by selecting a few representative papers from the literature. And I hope what we can learn by that is both what we've done today, and we can start to think about how that may project forward into what potential achievements or applications of synthetic biology might be in the future. Unlike George, I am going to start with the definition. You've heard a lot of them, and you've heard, I think what's clear is that there still is, I will say, a lack of universal agreement on what synthetic biology is and how it should be defined. I'm going to give a practical definition, one that we use within SynBERC, the Synthetic Biology Engineering Research Center. It's very simple, and it's a goal-oriented definition which says that synthetic biology is about making biology easier to engineer. You've heard some of these things before, particularly this morning in the first session about the relationship between biology and engineering and how those may interact with each other. For us in particular, this is about applying engineering principles to biological systems, and this involves words like design and modeling and characterization. I actually was raised academically by Jay Kiesling in the metabolic engineering community, and there's a well-known cartoon that Greg Stephanopoulos at MIT used to show where there's a group of students in a room, and the first day of class, and the student raises their hand and says, what's the difference between metabolic engineering and genetic engineering? And the next cartoon panel is a picture of the professor that says lots and lots of math. And the third panel is an empty room of just the professor, and all the students have gone away. There is very much within engineering this idea that we like to have models of systems that are numerical, they are mathematical, and it's an attempt to understand how the systems behave so that we can have this loop of design and modeling and characterization to loop back into a redesign to change your model and see what the new characteristics are. And so I think that is a part of synthetic biology which has traditionally been different from genetic engineering, but is not wholly distinct from what you may know as systems biology. Again, this effort to include math and the ability to predict and design and what we do. I will highlight DNA synthesis as an enabling technology, but you'll see from the first few slides that if we're talking about making biology easier to engineer and we want to get started with that now, and based on, I thought, what Drew gave us a very good slide of the technology gap, if you will, between the ability to write DNA and to know what to write on DNA, much of what's happening now under the umbrella of synthetic biology is using DNA synthesis at a very minimal level because we have to start with some existing biological substrate. So in that vein, if we think about this goal of engineering biology, what is our biological workspace? We heard a lot about microbes being a cellular substrate of choice because of their relative simplicity, and I'll use relative there quite intentionally because we're still talking about very complex organisms, even though they are less complex than mammalian cells, which you see there, and also plants. And if we think about now from an applications-oriented perspective, if these are the biological substrates that we want to work with, then the applications, I think, become pretty clear in terms of extrapolating that we can think about therapeutics. That includes pharmaceuticals in terms of traditional small molecule pharmaceuticals as well as biologics, or what's referred to as biopharmaceuticals, essentially protein therapeutics or more complex therapeutic agents. Energy, especially fuels, but not restricted to fuels, and I'll give a brief slide on that. Chemicals, which may be part of the pharmaceuticals, but also increasingly is leading towards thinking of new ways for materials, to have renewable materials, things to get rid of our polypropylene bottles, which will fill landfills if we can't figure out good ways to recycle them. And then agriculture, as we go from plants, think about the biological workspace of plants, then we really are talking about the potential to extend even what we've seen before with genetically engineered or genetically modified organisms for agriculture. Wrong button. Too many buttons. With this paper here that we've heard about already, which is the work from the Kiesling Lab at the University of California at Berkeley on producing this anti-malarial drug, artemisinic acid, which can be chemically converted into artemisinin for use as an anti-malarial. This was funded, as the numbers have come up over and over again, so I'm sure we can all recite them by now, $42.5 million from the Gates Foundation, as something of a public-private partnership between UC Berkeley and Amyris, which was a company founded by folks from the Kiesling Lab to develop this technology. One of the unique aspects, intellectual property came up previously. There were lots of issues because the University of California had to agree to make the licenses available essentially free, and the commitment by all parties involved is that they would develop this as a remedy for at cost production. So this was to be a non-profit generating venture as far as the company is concerned. Amyris, if you've been keeping up with the literature, has sort of transitioned this process. It's now in the hands of an industrial manufacturer, and they've switched their focus almost exclusively to fuels. So it's an example of how the basic technology of these achievements and what we're able to do with engineering of biology, with synthetic biology, with metabolic engineering, whatever particular phase you want to use, builds a repository of intellectual information and intellectual property that can be then converted into other downstream applications, and in this case, again, from therapeutics to fuels. We've talked a lot about microbes. That work was all done in microbes. There are efforts and achievements in synthetic biology that are going into increasingly more complex systems. This is a paper from Martin Fussenegger's group at the ETH in Zurich, from PNAS from a year ago, about now developing effectively a circuit to be able to control gene expression for implants. So the idea is that they were able to take pieces from microbial cells in order to put together a regulatory element that would respond to a particular molecule that they then put into a skin lotion. They could have subcutaneous implants, and then if you applied this lotion, you would get gene expression. So from, again, this notion of a circuit where you can control design or control expression of a gene from the introduction of a small molecule, this is now an example where we can think about how that actually has potential applications in medicine in terms of being able to activate gene expression, perhaps with novel forms of gene therapy, in a way that would now be a subcutaneous implant. So you're not talking about trying to modify the genome with more, I would say, complex perspectives of gene therapy where you're looking at, for example, removing stem cells and re-engineering them and putting them back into the cell. This would be a separate implant that would be distinct from the native or the human chromosome. Moving on to fuels, I've already mentioned Amyris's work. This is work from Jin Liao at the University of California at Los Angeles that was published in Nature a couple of years ago on new pathways for making higher order branched alcohols as biofuels. This is technology that's been licensed by Gevo. So there is, Dr. Bassler this morning mentioned that one of the differences was, and if I can paraphrase, going from more of a proof of concept to these ideas of scale and optimization and really getting something industrially viable. And this is a case now where this work was licensed by a company, and they're actively working to commercialize this process. Similar to the work done with artemisinin that we've heard about before, these were pathways where, to a certain extent, they're all natural. The molecules that were being produced were ones that had been identified as minor products in wine fermentation, and so the enzymes or the genes needed in order to convert what ends up being intermediates in amino acid synthesis or amino acid biosynthesis into these alcohols were optimized in a way that the most promising in this case was about 22 grams per liter of isobutanol being produced. And then LS9, Dr. Church has already referred to, and this is just a screenshot from their site, from their website. And I wanted to highlight the fact that they really do talk about themselves as being a synthetic biology company, being able to take advantage, as someone has already referred to, of all of the extensive information that's come to us from genome sequencing projects, but increasingly the tools and technologies that we're developing and being able to take advantage of that have to do with synthesis and construction of biology. And they are focusing on fuels, but also on biochemicals. This is an example that I mentioned before in terms of energy but not being fuels related, and this is a very recent paper just in April of this year from a lab at MIT where again, because of the multiple definitions of synthetic biology, we may or may not think about this as synthetic biology. But to just describe briefly what was done here, the Belcher lab at MIT uses engineered phage, M13 phage, as biotemplating devices. So they were able to use them to form these self-assembled structures, and the phage actually interact with inorganic often metals, and they're able to form these higher order structures. So this is a case now of using biological inspiration, if you will, or using biology as a template for making these nanostructures. But we could certainly think about how to expand that towards now having the power of synthetic biology and constructive biology to be able to redesign these phages in different ways so that the types of structures we get are ever more complex. And the particular application in this case was to be able to put together a structure that would allow you to have effectively photosynthesis. So they had light-driven water splitting with the idea that you could use this for energy storage because you could capture now the hydrogen that comes from the splitting of water, and that hydrogen can be stored and used at a later time. Whereas if we think about traditional solar energy, then you have it available when the sun is out and you don't have it available when the sun is not out. This is work actually from my own lab in collaboration with John Duber and Jay Kiesling in the chemical space, where here what we were looking at was being able to make now a pathway for a compound called glucuric acid, where we don't actually have a natural metabolic pathway for this compound. And this is a little bit different from the work I presented previously on artemisinin and also on the branched alcohols from the Liao group, where we weren't starting from a pathway and then trying to reconstruct it. We actually started from, here's a compound we want to make. How can we think about doing that? And the particular synthetic biology innovation in this case was to be able to use these novel synthetic scaffolds. And Dr. Bachelor this morning mentioned the wonderful spatial organization that happens within naturally occurring systems. And this was a synthetic device that was designed to introduce this kind of spatial organization into a microbial cell, and the result of that was to have increased productivity for the compounds we were interested in. And then finally, I wanted to sort of wrap up with a few examples from what's increasingly referred to as biological computing, or from a lot of the analogies to programmability. The first of which was a paper about 10 years ago now that described the repressilator, which is essentially an oscillatory network. And the instances of these have only grown a few years later, also from Jim Liao's group. The first was from Princeton, and Mike Elowitz is now at Caltech. This synthetic gene metabolic oscillator, which was called the metabolator, and it was essentially an oscillator that, rather than oscillating for a specific gene output, which is often a fluorescent protein, this was oscillations in the levels of a specific metabolite. Now going from, again, microbial systems into mammalian systems, again, the Fuseniger group just last year reported on oscillators in mammalian cells, and then this last paper was referred to this morning by Dr. Bachelor as well, as now looking at these oscillators, genetic clocks are referred to here, with taking advantage now of intercellular communication. So this was a synchronized community of these cells that will respond in these ways. And when I think about these particular applications, these are often discussed, sometimes derided as toy applications, and all you're doing is making cells blink. What is that really good for? From my own perspective, as someone who's interested in trying to make these systems that can produce high quantities of synthetic chemicals, then we're interested in these because we know that timing of gene expression is important for some systems that we're looking at. And so we can look at oscillators that have been designed even with gluing fluorescent proteins and think about how do we extend those into practical applications of systems where we're using them either in therapeutic purposes in order to have timed expression of genes, for example, in development when we're talking about stem cell biology, or even in again, a large bioreactor where we're ultimately talking about chemicals. The last sort of screenshot I have is the paper that, again, was sort of the impetus for this particular discussion from the Venter Group, which you've already heard about. And my comment I wanted to make here is that all the things I've talked about so far, you may be thinking, well, what does that have to do with synthetic genomics? What does that have to do with the ability to actually completely synthesize a copy of a bacterial genome? And what I would say to that is that this is about trying to bridge this technological divide, that what we currently have is the capacity to do very extensive re-engineering of genomes from existing cells, taking out lots of genes, putting in lots of genes, but beyond that, where the challenges often arise are how do you precisely control them, temporally, spatially, all these other issues about natural biology. They're very complex and very confusing for us. And what you have here is now this very clear synthetic capability. And where I see this bridging is that as we get better and better at understanding how to do the kinds of engineering we're doing, then it really is about the differences in scale that Dr. Bassler referred to this morning, that we can think about now going from making these manipulations at the level of an existing genome towards designing them in de novo and starting from scratch with a genome that works the way we want it to work. So the final comments I'll make is that there are, of course, lots of challenges. Biology is complex, as we've heard over and over again, and I'll add that it's often context dependent. So we do have this dream of having interchangeable, interoperable parts. I'll say from personal experience, you move them from one cell to the other, they don't work the same way, okay? And that's exciting. It's a challenge. It's something that we have to become better at understanding. The synthesis capabilities, as you've already heard, far exceed the design capabilities, and that's a technological gap that does in some way point to what our future ambitions are, but indicate what our current limitations are. The potential benefits, I think, are enormous. I've indicated a few of these, but we can think about this in any way where we can think about biology being important. At the same time, the risks are real. Because there is this information gap between what we really understand about biology and what our capabilities are, it's impossible for us to really predict what's going to happen in every single experiment. And so I do think it's very worthwhile to think about being as careful as possible as we do this to minimize those risks. And 2 seconds over, I'll stop.
That's very impressive. No, thank you very much for that list and also ending with the challenge that we have ahead. Keeping with the format that we used before, I ask the commissioners to get their thoughts together, but I'll return the favor. Amy, if you would like to offer the first question.
Thank you very much, and thank you all. Let me begin with a question to Craig, if I may. The potential power of synthetic biology creates hopes and it creates fears, and we're all too well aware now of the fears, but I want to begin with the hopes as well. So you mentioned a one-day production of a vaccine for a flu, for example. So here's my question to you. What is the single hope that we should most believe in from synthetic biology moving forward? And it would only be incumbent on me to ask you the same question with regard to fear. What is the single fear that we should take most seriously?
Well, they both give me wide latitude, so I appreciate that. So I think...
Don't make it too wide.
Yeah. On the hope side, obviously what our own teams and others are trying to do as well, we need new tools to make new medicines a lot faster, particularly with vaccines. It took quite a while with H1N1 to get a proper response, in part because the rate of building and deciding on seed stocks, and in part because we're using 100-year-old technology with chicken eggs to produce vaccines. Both need to change and quickly. But with rapid sequencing and all these changes in reading the genetic code, and now the ability to quickly write the genetic code, it's now hours instead of weeks and months to make new seed stocks. The potential applications, because we can design cells with hundreds to thousands of antigenic variation, diseases like HIV that we were chatting about that change their genetic code very rapidly, things like rhinovirus. Well, we don't have a vaccine against the common cold because the virus evolves very rapidly. Designing things either with the same rate of evolution or covering the spectrum of antigenic variations gives us whole new ways to approach vaccines that never existed before. On the environmental side, I think it's clear we need to do something different than the environment as we go from 6.5 to 9 to 10 billion people. We can't keep doing what we're doing. And so attempts, all these different attempts, they all need to be successful in creating new sources of fuel and energy and food, or humanity will be irreversibly damaged and altered. So we are a society dependent on science now for our future. Biology is a key part of that future science. Synthetic biology, synthetic genomics are key, I hope, components of altering that future. On the fear side, obviously, the worst scenario is what happened in computing, because we're talking about software. People make computer viruses that cause a lot of economic damage. We don't want the same mentality going into making new animal or plant viruses, either inadvertently or purposely. And some of that can be readily prevented by some pretty straightforward regulations. But obviously, nobody who develops new technology wants to see that ever produce harm to others. We just would like to see just the benefits. I think the molecular biology community has a pretty good track record for the last several decades because of the guidelines and rules that we've all been working under.
So I also want to direct this question to Dr. Venter. I've heard both you use and in the literature people have talked about the publication in Science as a proof of concept. And I wanted to understand exactly what it is that it is proving, in part because, as I understand it, the cell wall of the bacteria was used in the first generation, and it's a natural organism that has been synthesized. So I'd like to understand what it is that it proves and how significant that proof of concept is. But second, building on that, looking forward, I understand that you may be working on algae and other multicellular organisms where the genetic information is in the nucleus of the cell rather than as a single strand. And I'm wondering how far away are we from that, and is that really the kind of proof of concept that will propel this field forward?
Okay. So what's been possible in molecular biology is what several people have described this morning, changing one or a few genes in the cells by inserting those genes in plasmids. While we have evidence in evolution that many bacteria evolved by taking up entire chromosomes, and for example, cholera actually has 2 chromosomes from 2 very clearly different origins. So they probably happen through these kind of processes. But never before have we molecular biologists been able to take an entire bacterial chromosome, an entire chromosome of anything other than a small virus, transplant it into a cell of one type and convert that cell into another. Then you add to that, starting with the digital code in the computer, making the entire chromosome from scratch means now we have the means to start with that digital code, make dramatic changes. I mean, while we had the basis of an existing organism, we made substantial changes to it. We inserted the names of 46 authors, several quotations. It's the first genome with its own built-in website, web address. These may seem like trivial changes, but they clearly identify it as a synthetically made chromosome, something we think is critical for this field, and activated that and completely transformed that one cell into a new cell. It was not trivial. One base pair being wrong set us back 3 months. So one error out of over 1 million base pairs did not enable this to happen. So it's now, because it's a proof of concept, we do know how to do it. And now we can make much more extensive modifications. So we're building a robot to do combinatorial synthesis, where instead of making one chromosome over 10 years like we did, our goal is to make 1 million or so a day by randomly sorting genes or selecting the very specific ones, selecting for living cells that you can't get. It's not a species that existed. It's very closely similar to a pre-existing cell, but it grows substantially faster because of the 14 genes we eliminated.
And on the multicellular front?
You can have a nucleus and not be a multicellular
Sure.
system. There's a lot of single-cell eukaryotes, and a lot of the algae are in that category. Moving nuclei around has been done for, 50 years or more. Changing the DNA in the nuclei and replacing the DNA and moving those around, we don't think will be a huge challenge. And in fact, it's probably easier to replace all the chromosomes in the eukaryotic yeast just by replacing them one at a time with synthetic DNA.
Thank you.
So, Dr. Ventnor, do you think it's fair to say that, in the very elegant transformation experiment, that really, that's how when I read your paper first on that day, I saw it as, probably the world's most elegant bacterial transformation that had been done to date.
Thank you.
But I think maybe trying to clarify what Nita was driving at, you really needed to collaborate with existing life in order to make that transformation experiment work. And while it's true that after several replications that all components, not just the proteins of that cell, were derived from what you had produced in silico and then printed out, the initial step did require a collaboration with existing life that had been derived by natural selection.
Yeah, absolutely. So we're starting, as I said, with the 3.5 billion years of evolution. we use that starting system to read the new genetic code and start making all the new proteins. As I said earlier, I think it's an interesting scientific question how few of those components we can get away with. As I said, perhaps just a ribosome, some polymerase, some tRNAs, a few lipids. When people evoke that you start with existing life, it takes us back to vitalism. That people try, and amazingly, the New York Times has tried to re-evoke vitalism. Most scientists view that it disappeared 80 years ago as a concept, and certainly with DNA being the genetic material coding for everything, there's nothing vital in the cell other than the ability to read that new software. So we are clearly software-driven machines. That software is DNA.
First, thank you all very much for your comments. I would like to ask Dr. Venter, this is an important scientific step, but as you described what you've been working on for many years, you also described a process of thinking about the ethical issues right from the very beginning. So I wonder if you could say, from your perspective, what has changed now ethically, if anything, and where you think, building on Dr. Atkinson's question earlier, where you think I think you mentioned we need an intelligent ethical legal framework. So where are we lacking in that regard, or what do you think we can do to help in that regard? And I'd love to hear others' opinions on that as well.
I think it's a very critical question. It's not clear that anything has changed so dramatically as what some people were describing as minor changes in biology. We have minor but significant changes in the ethical and legal framework, primarily because the way we control who has, for example, A-list agents has been controlling who has access to these agents. Now, if all you need is the genetic code in the computer, it totally changes who has access and how you get access to them. If students can order anything from a DNA synthesis company and there's no tracking of what they order, somebody could try and make Ebola virus, which is only 8 genes, or at least the DNA. The DNA is not infective, but I'm sure if Homeland Security started detecting Ebola virus DNA, they'd probably get a little upset. Those would be the kind of hacking things that we don't want to occur. I think these can be pretty much eliminated by requiring companies to screen against A-list agents and requiring bona fide institutions to be doing this work versus being done in somebody's garage. I think creating new life forms, I think what we did is as much a philosophical step as a scientific technical one, because it now opens the window for literally merging the digital world with the biology world. And because anything that's totally open-ended, we think there's some guidelines that are needed. I think it's sensible to start in that framework so that we don't get the negative consequences or the unintended ones from lack of paying proper attention.
George, you've written on this as well. Would you weigh in, please?
Yeah, I'm not sure whether this is an ethical or policy issue, but many of the previous discussions the conclusions have been we should have more discussion. And I actually think that we're actually in a place that we can do more than that, which is to focus on licensing and surveillance. And I don't know whether that's ethics at all, much less a new one. But what has happened since 1999 is this exponential curve has gotten steeper, and I think that's something you can't ignore. So I would say that it's time to go beyond having more discussions.
Following actually on that, it seems to me we've heard a lot, both from the previous panel and from the three of you, about the widespread availability of various codes, the quote-unquote ability to do it in your garage, unquote. But that seems to me to refer to the obtaining of the sequences and perhaps the synthesizing of those. And that generates worries for people. But the question I have is how big a step is it, and you've alluded to this, I think, Dr. Venter, from having the sequence to actually getting it to work in a biological system, and is that gap big enough that we shouldn't be as fearful as we are of the possibility of this being misused, because we could, in fact, have regulation or safeguards at that step that would be very helpful? True or false?
That wasn't a yes/no question. I'm sorry.
You have a 50% chance of getting it right.
Then I'll say it's false. I think with each new cellular system, and by the way, the mycoplasmas don't have a cell wall, and that was one of the reasons we chose it. They have a simple plasma membrane, which made it pretty simple to get the DNA across. But what we're trying to do at the Venter Institute with synthetic algae right now is maybe using spheroplasts, which have a plasma membrane, and you can transform things. I mean, we're at the earliest stages. We need to see how extendable these tools are. Getting DNA past cell walls may be very tough, but there's other ways to get around things. So the two areas go in parallel. One is the design and the synthesis, and the two booting it up. Our biggest worry was we were going to have this really nice macromolecule, the largest one of a defined structure ever made, and we couldn't activate it in a cell. And we were there for a long time because of one single error in the genetic code. So I think it's going to have to be optimized for each individual biological system. It's totally different getting DNA into plants than it is to bacteria, totally different with cell walls, without cell walls. But I think this is going to be a very rapid expanding area of research and probably difficult to regulate. I think the guidelines that get set up for approving projects at the institutional level with broader guidelines at the funding level, and even though our work was not federally funded, because my institution is a major federal grant recipient, we have to follow the federal rules regardless of whether it's funding that particular research. So I think the way molecular biology has been practiced, particularly in this country, has been, I think, a wonderful example of how to proceed, but expanding the repertoire and expanding some of the ways we monitor things.
And Dr. Prather, you had, go ahead.
Yeah, so I may be misinterpreting the question, but so the information is free. We can go to the NCBI database and get as much sequence as we want. It is cheaper, but still not trivial to actually pay for synthesis. So my lab does not yet, as a matter of practice, synthesize everything. We still do a tremendous amount of PCR. I just had a meeting with a student a couple days ago and said, okay, you can get these things synthesized because it's going to cost about $3,000, but you can't get the 12 other variants of it synthesized that you want, right? Because now we're talking about $36,000. So as far as access, some of it is thinking to the future in terms of if we go, certainly we're not at $10 a base, as George showed for synthesizing the first plasmid, but we're under $1 per base, but we're not yet at a dime per base at the level of small amounts of orders. Okay, so you can do negotiations with some companies to get things on the order of 10 cents to 25 cents a base if you want a lot of sequence. So because of that, I think, and again, I may be misunderstanding your question, but I think there are different answers in terms of whether or not you're talking about institutional access versus non-institutional access, skilled labor versus unskilled labor. So in terms of access, because of the costs, I still think that a lot of the more fear was a word that was used earlier, but the things that may invoke a lot of fear and apprehension are still beyond the cost of most non-institutional players. And then, because we're really talking about difficult biology and one base pair mistake setting you back 3 months, there's still a big difference between what you can do as a skilled practitioner versus an unskilled practitioner.
Just to follow, I appreciate hearing that it's a little more difficult than just doing it, quote unquote, in your garage to get the sequencing done. But I was talking about the next step and whether people can do that in their garages, quote unquote, of getting that to replicate inside a cell and how difficult that is and what material is needed there and whether that's an important place in which regulatory safeguards could be placed to make sure this doesn't get into the wrong hands.
So at the simplest level, let's say, and if you read some of the blogs and the popular press, everybody wants to make things glow. We want fish that glow and cocker spaniels that glow, and so it's like, let's put green fluorescent protein in anything we can think about. It is relatively inexpensive and, on a skill level, relatively easy to order a gene that would effectively be a plasmid that encodes for green fluorescent protein with a promoter on one end and a terminator on the other end, and to transform a very simple bacterium with that in your garage and say, hey, it glows. It's very difficult to make your dog glow. Right? So again, we're still talking about a level of complexity there. And so one gene, being able to transfer one gene and getting that to work in a garage with a junior high school student, pretty close to trivial. The types of things that the Venter Lab did, not going to happen in a garage with 14-year-olds. But very expensive. Yeah.
Thank you very much for your presentations. And all three of you really talked about the need for some level of regulation. And I wonder if you could comment on whether all of these different things that would fall under the definition of synthetic biology are already covered under existing regulations, because certainly we have regulations about how to handle anthrax or Ebola or other types of things. Or do you feel that we need to have new and different types of regulations to deal with the issues of synthetic biology?
We certainly have recombinant DNA regulations. Many of these depend on the person practicing it having federal grants or in some other way being a responsible citizen. I think what we don't really have is surveillance that the regulations are being obeyed by all citizens, not just the standard members of society. And I think we also don't really have many regulations about safety testing as we make things that either are intended or could accidentally get into the environment. I think as safety testing we take for granted in many other engineering disciplines. There's relatively little of that in biology. It probably doesn't require major overhauls, but I think there are some gaps that we need to pay attention to.
There are really no limitations on what you can order from an oligonucleotide synthesis company. At present time, they're not required to screen against any list of agents. Some are voluntarily doing it now. And it's not just a U.S. problem. DNA synthesis is a global effort. If you can't get what you want here, you can order it in Germany, or you can order it in India, or get things made in China. You can buy DNA synthesizers off of eBay. So maybe, but there are, as people said earlier, 4 companies that are probably 90% of the synthesis in the U.S., even though they're not all in the U.S., requiring them to screen against A-list agents, requiring them to have bona fide credentials of the ordering institution, I think are things that could go towards preventing the frivolous use. There is a lot of homebrew biology being done in kitchens. It's a new trend. Drew Indy's been, in the past, I was pleased to see that he stopped doing it, encouraging biohacking. We want some reasonable restraints on that without destroying this wonderful creativity that these kids are doing to come up with some new circuitry that could totally change what we work on. But I don't think it's covered by any of the existing regulations.
But just as a follow-up, in the kinds of experiments that you published, it is possible to be able to take 100 MERS that nobody might be able to recognize that they may come from, a pathogenic organism, and you could order a bunch from one company and a bunch from another company and be able to put together.
Yeah. So at the level of 100 MERS or even anything over probably an 18 MER or something, you could get a pretty good trend of what somebody was trying to do. I mean, the signatures are pretty clear-cut.
Also, these companies are beginning to coordinate voluntarily. This is something that would be nice to be backed up with a regulation, but they're voluntarily coordinating their efforts. So if someone split their order out over 4 companies, that in and of itself would be an alarming event. Which combined with the sequences that can be recognized, I think you could put the story together. But there will be ongoing efforts to get around that.
How quickly would you put the story together, realistically?
Well, if it's entirely based on computational algorithms that have been pre-tested, and again, I emphasize the importance of testing, then you could put it together in hours. The computers, especially if you've got government agencies that are willing to act in hours.
Yeah, I think you've gotten to the heart of a lot of my concerns. As you probably all know, the Amsham-Riccio cult had an avirulent strain of anthrax. However, that seems to be becoming a low hurdle to overcome in the ongoing biological processes. So I've heard a couple of different things. One is that this is still difficult to do, very difficult to do. But the second part is that it's getting easier. And so I think that Raj brought up an excellent point, is that there are regulations in place now for, I think, what we would consider traditional biology, being able to reproduce organisms that are on the biological and select agent toxins list. However, we're talking more about the biobricks now, which, quite frankly, are not part of the regulation for BSAT. And so what concerns, and I think you've expressed them here, so your concerns about this evolving technology in getting around the BSAT, the security measures that we would need to take to make sure that these would not happen, and what is the balance between, you've probably been privy to the discussions of the latest BSAT, in balancing scientific discovery versus the security for the American people?
I don't actually think that this is a trade-off between security and scientific discovery. I think if this is properly implemented, where most of the effort is in developing computer software and getting compliance at the company level and getting surveillance at the government level, the researchers, in a certain sense, shouldn't even see it. Should be transparent to them, and they can get on with their work. On the other hand, if you require them to sign a piece of paper every few minutes, every time they pipette something, then you could interfere. I think it's unlikely that that's where we would be going with this, but I think some serious computational efforts are in order.
I'm interested in the money behind it all. It's a very expensive proposition now to come up with a new cell, as you did, over $40 million, or new products, and mostly funded now in often small biotech companies and with venture capital kinds of money. Are there recommendations for being able to encourage the entrepreneurship while not having so tight control on it that you can't get a payback to the amount of money you spend putting into a project and can't get that payback fairly quickly, versus being able to also encourage entrepreneurs to work on projects? I mean, there's going to be huge profits in this if it would work out the way it is looking like it might work out in biofuels or energy and so on. And I just wondered if you had recommendations or thoughts on what the Commission should recommend on those issues.
Just my opinion is where this current system is actually quite healthy. In contrast to the one that Rob Carlson described, that anecdote, most of my experience with dozens of companies is that they can get the job done without spending a lot of money on lawyers. Very often you don't really even need the patents. In the end, it's the know-how that's really incredibly important. I have very few examples of a patent getting in the way of academic research, and generally not even getting in the way of startups as well. This is such a vibrant field that people are inventing so quickly that they invent around or don't even concern themselves with it. So I actually think it's quite healthy, and going from small to large is happening quite quickly too. I mean, Craig mentioned Exxon. In the case of LS9, they've got Procter & Gamble and Chevron. This is all in very short number of years. So I really think my opinion is it's healthy.
In fact, if I can just add briefly to it, it's healthy and critical. I think if all these bets are right that everybody's placing to get a proper ecological benefit and change the use and dependency on taking carbon out of the ground and burning it, putting it in the atmosphere, we need things to work economically. And I think it's very fortunate that there's a healthy investment climate in the U.S., despite the recent changes in the stock market, that have stepped in where the government hasn't. Most of the advances in biotechnology have come starting with companies like Genentech and with private investment going forward. I'd say the majority of them come from that. And in our own case, we would have been stuck back in 2003 with a small synthetic virus if we did not have independent money from starting synthetic genomics to actually fund this work at the not-for-profit institute.
I would only add to that I think I agree with what's been said. I think one of the impediments to progress, if you will, that can arise if all of the achievements are done individually is one of the very big goals of synthetic biology is to have standardization and interoperability. And so one of the ways where I think the federal government can help with that is to promote in some tangible way an effort for the community to be able to organize on a regular basis around what should those standards be, so that you don't have innovation happening in isolation, in a way that you have very great technologies evolving independently, and then to network those or to interface those becomes very difficult. What we always think about as we dream about synthetic biology, and you see the Lego kits all over the place as a good analogy. That works because you do have standardization, and you know that you can get Legos from anywhere and they're going to work together. And that's an effort that I think has been more difficult to get real support for, because it's fundamental, it's foundational, and it's enabling, but it doesn't in and of itself get you biofuels. And it doesn't in and of itself get you new vaccines, right? It facilitates all of those things. And sometimes there is a gap between the foundational, more engineering-oriented standardization work and the applications-oriented things, which can be very interesting and attractive to investors.
Yeah, I want to begin by thanking all of you three for excellent presentations. I've got a couple of concerns or questions for you. One has to do with the fact that this is all now accessible on the internet and it's international. So if we're a commission set up to think about regulations here in the United States, I'm wondering what the context of our deliberation should be if these activities are really taking place all over the world. So, what sort of international collaboration has to take place for U.S. regulations to have any real effective bite? That's the first question.
I think it's a critical question because science is international, these tools are international, the internet is international, and I think first and foremost the U.S. can set a good positive example. That didn't happen with stem cells in the recent past. And research expanded overseas at the expense of research in the U.S. I think we can do the opposite here if we do it intelligently. The same concerns that we have here have been expressed in the EU and basically every country I've visited around the world. So I think if there's a positive example of how to deal with things, that would be a good start. But it has to be international ultimately to have any impact.
Yeah, thank you. A follow-up it's really a follow-up question about the role of industry. There's been some talk around the table about the movement from small to large, right? So listening to all the panelists, you get the picture that we're currently living through an era of a kind of biological Woodstock, right? With people experimenting in their garages and so forth. But the movement will be, as it's been in the pharmaceutical industry and computer industry, from small to large. And so I'm wondering what the implications of that might be with regard to access to the goods produced by this industry. We've seen in the area of pharmaceuticals a lot of public concern about the patent system and the rules and regulations relating to access, particularly with regard to access to life-saving drugs for diseases like HIV, where it's perceived by many people that the patent system is working against access to life-saving medications. So I'm wondering if we have anything to worry about that's analogous in this area, right? In other words, should we be worrying now about the synthetic bioanalogs of Microsoft and Pfizer limiting access to knowledge and limiting access to the goods that are produced?
My answer is quite simple, no. I don't think there's any worry at all. In fact, the worry is in the opposite direction. If we don't get things that really work at a commercial level, this is an interesting academic field. Publishing a paper like my team did in Science is great for understanding the concepts, converting it into reality where you can buy fuel at the gas pump made from carbon dioxide instead of from oil out of the ground will only work if that's done in an economically competitive environment. LS9, all these companies, synthetic genomics, they'll only survive if they have economically competitive products. Most people aren't going to buy things just because it's better for the environment, unfortunately. So any new fuels, for example, have to be available and they have to be cheaper than existing fuels, or at least cost competitive with it. So we need economic driving forces to pull this stuff much more rapidly than is currently happening. I don't see any limitation of access. We need access pretty rapidly to CO2-based fuels, as an example.
Before I go to Nita and Anita, I don't want a thought that was dropped, Chrysler, that you brought up and connected to something you said, George. It's the flip side of regulation. We've been talking a lot about regulation, and that is stimulation. Well, one of the effective ways to ensure safety to be in sort of a Gretzky-esque way, be certain that we have the ability to skate ahead of the puck, where the puck is going. Were you suggesting, Dr. Prather, that or maybe I should just leave it open, the question that we had sort of posed in the prior panel to you, what would be the very next thing to be funded? Where would we like to see, in view of being able to have a knowledge base and an ability both to advance the applications of the kinds you've all been talking about, but to have a deeper knowledge to help ensure that we can recognize, as Dr. Church mentioned, some of the potentially sinister applications of these things. What would you fund next? Would it be your standardization?
You're not allowed to answer your own lab.
Yeah, fair enough. Okay, what's the second thing you would fund?
No, it's a difficult question. So I think about, for example, the BioFab, which Jurindi is directing, which really has a very ambitious goal of being a focal point where you can develop parts, to use a term in synthetic biology, so discrete pieces of DNA that encode for some biological function. And you can characterize them, and you can understand them, and you can see how they behave, and you can study things like composability of what happens if I put this thing with the other thing. And I think it's a very ambitious goal, and I think it's great. And they've got like $2 million in 2 years, and so what happens when that's gone? Right. So I think that there's a need for an effort that is more ambitious in scope and much bigger in scope to say, okay, let's bring, that does two things. One is that it can serve as a forum, if you will, for bringing different players together and brainstorming and saying, okay, here's what I'm doing, here's what you're doing, here's what this person is doing. How do we get those to interface in a way that we can actually set a standard moving forward so that as new technologies are developed, we know they're going to fit in very well? And then how do we set priorities for, and safely, yes. And so, that's, I'll jump ahead to a thought. I think one of the things that's been very nice about the Stenberg experience is that we've had all along discussions about, we have a thrust which we call human practices that deals with biosafety and biosecurity and intellectual property and societal implications. And so, but that's a very, very small number of people in what's becoming an increasingly populated academic field. If you look at the number of people who are associating themselves with synthetic biology, it's grown astronomically over just the past few years. And so there are questions in terms of if you were focusing, if you take NSF's investment into SINBRC and you take DOE's investment into JBAI, the Joint Bioenergy Institute, you're talking about, real money. We're not going to give it back, right? It's not trivial, but it's a very small number of people that it's impacting. And so what I'd like to see are efforts that are designed to bring the community together in a way that we can think about what the next steps are going forward, and that we can be more progressive and proactive as opposed to reactive and saying, well, you did it wrong, so here's my other way to do better. We're in that midst now where it's a bunch of, you go back and forth between I did it this way, that way is all wrong, here's why it's all wrong. And I'm not saying that won't eventually get us to where we're going, but if we want to be able to bridge this technological divide and we say we have really exciting technology and the potentials for the impact it can actually have on human existence are very real, we need to be able to move that forward more quickly.
Thank you. Craig, you have a comment on this?
So the two questions I get most often when I give lectures on this topic is people are worried about bioterrorism and environmental release. And it depends where you are, which one's first or second. And so George gave some wonderful examples of various safety mechanisms that could be built in, but it'd be nice to have orders of magnitude more. We're trying to build in suicide genes to organisms. If we're going to have large algae plants made from genetically synthesized or modified organisms, they need to not be able to survive in the environment on their own. Suicide genes, chemical dependencies, using artificial amino acids so they couldn't possibly grow in different environments. Expanding the repertoire of what's safe and secure, I think, would be the single most beneficial thing out of any government funding.
Just quickly, I think coupling this question to the previous one about small versus large, I think large actually has certain safety advantages. So it's only once we got to very large manufacturing of automobiles that we really started getting very high levels of safety. And furthermore, as the technology gets to a certain point, amateurs stop making it. So, I made a computer when I was young. I wouldn't bother to make one today. The know-how starts to fade away at the grassroots level, which is a mixed blessing, but from a safety standpoint, I think is incredibly important.
Thank you. Certainly, since we're taking so much of a lead from engineering, it made me very proud of my heritage, actually, today. But the one thing that has been demonstrated in so many physical systems is that it is far more effective to design in safety than it is to try to regulate in safety. So that was the basis for those questions, and you were very responsive. Thank you. Nita, I think you're next. Oh, Anita's next.
Thank you. I really have appreciated all these remarks. I wanted to ask Dr. Prather a very specific question about something that you said toward the end of your slides. You had a slide on which you made the intriguing point that our synthesis capabilities exceed our design capabilities. We know how, but not what. Could you elaborate? The reason why I want you to elaborate is because I'm wondering if that we know how but not what points to some limitations on the applications that may be forthcoming from this science.
It absolutely does. So simply put, this is all back to comments that Bonnie Bassler made earlier, that these are really complex systems that we're talking about. I don't yet know anyone in the field for whom their design works the first time they implement it. And the question is always, I describe when I'm sort of giving my pitch to first-year graduate students, they say what we do is to pick molecules we want to build, and then we have problems. And your thesis research is all about how do you solve those problems and what do you learn from solving those problems. And we often learn things we didn't expect to learn. Sometimes we run into problems and we go, yeah, we figured that was coming eventually. There's two different aspects of it. One is that biology, even for very simple organisms, is still very complex. And so being able to predictably know what's going to happen if you make even a single perturbation is difficult to do. And we are trying to make lots and lots of perturbations. So that's one aspect of it. The other part of it is that the type of manipulations that we're talking about doing are in and of themselves somewhat different from what we see in nature. We're mimicking nature, but we're trying to take natural components and string them together in ways that haven't been done before. And so there's, in some cases, just a lack of fundamental knowledge of how that's going to behave. And so there's a need for experimentation to actually have the observation that says, okay, this is what I observed when I did this particular configuration. Let me make 4 or 5 different variants and see those observations, and then put it on a graph and see if it's just a random set of points, or if I can then draw some conclusion that if I have these specific changes, here's the effect I'm going to get from that. What the Venter Lab group, I think, showed is that the capability, the capacity to go from sequence on a computer into something that is physical DNA is there. But if you ask anybody, okay, you're free to write 500,000 base pairs of DNA, what would you do? If most of us want something functioning, we're going to copy something that already exists. Because we just don't know how to make it all work together. And even the stuff that's working together, we don't know why it's working the way it's working together.
So how can we get better at knowing what, given our genius at knowing how? How can we get to the knowing what?
I think there's 2 parts of it, which also came through this morning. Some of it is more information about biology. So we don't, as an engineer, I have no desire to interfere with a biologist doing what he or she does on a day-to-day basis to uncover fundamental knowledge of biology. In fact, I applaud it, and we steal as much of it as we can with proper credit so that there's no confusion that we took it from someplace else. Right? So we need biology to have the freedom to continue an investment in biology to learn that. At the same time, and this was alluded to previously, because we now have these synthetic capabilities, we have the possibility of doing kinds of experiments that we didn't have before, and it's mostly at a pace and a scale that we couldn't access before. So we can use the tools of synthetic biology to help us actually understand fundamental biology, to have rewiring, if you will, of a cell, and see if I make this perturbation, what does the change in the input tell me about the change in the output? And there are, again, numerous mathematical algorithms that have been developed and more that are being developed to help fill in the black box between here's your input that you changed and here's the output that you can measure. So there's and the other thing I think, honestly, that's a little bit more difficult, but it's about understanding. So there is, sometimes a little bit of lack of respect for biologists, for engineers, and vice versa, right? Engineers sometimes like to think they don't care about making anything work, and scientists sometimes say about engineers they don't care about how anything actually works, they just care about it working. So bridging that divide and having the understanding, especially for this field where they really are very interdependent upon each other, moves that forward as well. If we start to have conversations and say, here are the tools and techniques and methodologies I'm developing, how does that help you in understanding fundamental biology? And biologists saying, here are things that we've uncovered, parts as we would call them, what can you do with those? If we're able to have those conversations in a way that's respectful and appreciative of each other's strength, then I think the whole field moves forward.
I agree. No, thank you. Genetic deconvolution. Nina, last question from the Commission before we go to the open microphones. If folks want to queue up there, that would be fine.
Great. I just wanted to focus on a couple of the comments by Dr. Church and by Dr. Prather about the potential role for government both in regulation but also in promoting the developments in this field. In particular, I was hoping that, Dr. Church, you could expand on what you mean by surveillance. So you mentioned a few times that you think the government could play a role in surveillance, but I'm not sure if you mean that by active surveillance or passive surveillance or what specifically you had in mind. And Dr. Prather, when you mentioned standardization and that the government could play a role in standardization, I'm wondering what you're envisioning. Is it, excuse me, is it just funding, or should it be setting up sort of a large initiative like the Human Genome Project where it is something like a standardization project? So if you could expand on that as well.
So in terms of surveillance, I think one thing is just, I think it was hard for the government to act until industry had shown what it was planning on doing. But at this point, I think they could tune into that and help it along and make it law and work internationally. I know that both Secretaries Generals of the United Nations have been in favor of this. So it's a key time while there's still this bottleneck on synthesis. So and not just to regulate, to allow computer surveillance of orders of synthetic oligonucleotides and genes, but if you license the entire industry, then if someone wants to go around it, it's not just a matter of going to another shop or even going to an earlier step. They have to go around the entire system, obtaining the know-how to make the phosphoramidite chemicals all the way through to the know-how of getting DNA to work in a cell. I think that's a great opportunity, and I think it actually plays into what Christella will say. A genome project scale thing is I think, appropriate in this case. I was a beneficiary of the Human Genome Project, and I saw just how great it was in terms of stimulating community and industry. So I think there is a huge opportunity there that will also result in greater responsibility.
Thank you.
I'll give a very simple answer. You said funding or project. I would say both, which is basically to set a priority and say, we think this is really important, and so we're going to organize a project around that. And I don't know how you do projects without funding. So I think they go together.
Thank you. Are there questions from the audience? Yes, sir.
Terry Taylor from the International Council for the Life Sciences. My question is about the international environment. There's been an excellent discussion about the fact that anything we might do in the United States has to be fitted into an international context if it's all to be in any way successful with regard to regulation or ethical conduct. And I have two questions, really. One for our excellent speakers for first-class presentations is you're at the leading edge of the development of the science, whether it's in academia and of course experience in commercial industry. Where would you start in terms of the global environment? Is it best left to various networks which are academic, which are in commercial industry? Dr. Venter mentioned that industry is already trying to do some of these things, like the International Association for Synthetic Biology based in Germany, which is attempting codes of conduct and so on really quite successfully. Or should there be some central, more top-down approach? So is it a networking approach, multiple networks, or should we be doing something or encouraging the development top-down, centrally and globally? So that's question number one. Second question is really, I suppose, for the Commission, is to what extent are you going to consult internationally on this subject in order to set whatever recommendations you might make in a global context? Thank you very much.
Begin with the second one because I can answer that very quickly. We are going to consult internationally. Not only are we going to, we have already begun to consult internationally, and we will have here as a presenter somebody Marcus Schmidt, who represents an international voice in this, and we will continue to consult internationally. So I now push it back to our presenters to answer the substantive question, which is an important one.
I think the only way it can start is in two ways simultaneously, but it certainly has happened with molecular biology, an international code of standards that we all agree upon, such as simple things like labeling the DNA as synthetic by watermarking it, as we try to set the standard for trying to make sure no organisms are released to the environment unless they have these critical biological controls. We suggested early on no human pathogens, but 2 out of the 3 of the first viruses that were made were human pathogens, first with poliovirus, then with the 1918 flu virus. But the 1918 flu virus was done in the right way. It was done in the CDC with extreme controls and was a critical experiment for understanding why the 1918 flu was so lethal, because we couldn't tell that just from looking at the sequence. But I think it has to happen at the government and science society levels as well, in the key countries where these activities are going. Starting with the EU, China, India, and the U.S. would be great starting points.
I can't help myself here. As tempted as I am to wade into comments on licensing and regulation, I won't do that. But I did want to observe on the small versus large conversation that...
Introduce yourself.
Rob Carlson, previous speaker. That more than 50% of the jobs in this country are in small businesses. A little bit less than 50% of the payroll is in small businesses. And that's where a lot of our technology comes from. And there's already an example of small-scale distributed biological manufacturing being highly successful in this country. And if you look at the size of breweries over the last 100 years, officially the number went to 0 during Prohibition, officially. After Prohibition, brewing was dominated by very large-scale brewers around the world, actually, until 1980, roughly. And although a small number of very large brewers control most of the U.S. market, those number in the, you can count them on one hand, basically. There are 1,500 craft brewers in the U.S. They supply about 5% of the volume, but 9% of the revenue, or take home 9% of the revenue. So the point there is that is distributed biological manufacturing, and it works just fine. And they are covered by a regulatory regime to the extent that alcohol is still regulated in this country. That's all I wanted to throw into the discussion.
Thanks, Rob. Since you've brought it back to the Commission, Amy, you have a comment?
Yeah, I have a follow-up question. Since this panel, we really wanted to focus on applications. And here's the question. I'll try to make it as vivid as possible. Next flu season, the most virulent flu begins, and there is real fear of how much it's going to spread and the lives lost and so on. Next flu season, could we have a one-day production through synthetic biology of a flu vaccine?
Well, the seed stock for that vaccine could be produced in probably about 12 hours, and because all the surveillance now with the rapid DNA sequencing, we can predict, we think, well in advance what the changes will be for next year's flu before WHO even makes the decision as to the vaccine stocks. I think the biggest limitation going forward is how we actually produce the vaccine. Is it going to be in chicken eggs, or we're going to go to modern cellular systems that can do it orders of magnitude faster and under a much better control? So the second step is much more the rate-limiting step in producing the doses for the individuals. We're not tooled up. Well, Novartis and other companies now are tooled up for it, in part because of government funding, waiting mostly for FDA approval.
So if I'm right, the answer is no. We're not yet ready to do it in a day.
No, actually, I think we are.
Okay.
We are ready. Depends on who the we is. If we're the FDA, we're not ready.
So we're the public, we're the American public, and we want to know, next flu season, if there's some virulent flu strain, can synthetic biology come to the rescue?
So with what we're...
And I don't mean can it, if it were in theory possible, but will it happen?
It's very likely that, as I said, the vaccine you get next year will be from synthetic genomic technologies.
So the answer is yes.
The answer is definitely yes. NIH is already funding us to make synthetic segments of every virus, and it's easy just to put them together in a very rapid synthesis process to make any seed stock for any change we see for tracking new emerging infections.
Thank you for that.
There is a corollary to that.
Okay.
And the corollary to that question is that can it be done only by synthetic biology? Are there other approaches that could equally effectively produce the kind of vaccine that you talked about?
And what's the answer to that next flu season?
The answer is yes, but nowhere near as fast.
And speed matters.
Speed definitely matters.
And nowhere near as cost effective?
I mean, the cost is a trivial part of at that stage.
Great to know.
Just real quickly, sure.
I don't want to gloss over the manufacturing issues here because Dr. Venter made this point of if you're still making it in chicken eggs, it's not going to happen in a day. It's just not going to happen. So there's a difference between the tools of synthetic biology being able to give you what that starting material is. If we're stuck with chicken eggs, it's not going to happen. If you can go to chicken cell culture, it's going to be faster. If the DNA vaccine technology proves out and you can do it in microbes, you can absolutely do it in a day.
So what do we need for that?
That's not a synthetic biology problem. That's actually an immunology problem.
Right, exactly. Final question is yours, ma'am.
Nancy Jones from NIH. And so my question is, we've talked a lot about external regulations for the government in this, but as trainers of the future generation of scientists and engineers in this, what are your all's obligations? What do you teach your engineers and scientists about what the ethical considerations are for moving this field forward?
Well, at the Venter Institute in both synthetic genomics, we have internal IRB committees that any scientific experiments have to be approved in advance along guidelines published in the past by NIH, and also just a common sense approach. We wouldn't be testing new microbes to see if they survive in the environment. The mycoplasma that we made and changed was initially a goat pathogen. We've eliminated 14 genes that were supposedly associated with that pathogenicity, but we're not going out to goat farms and giving it to goats to see if, in fact, it still is a pathogen. I mean, there's sort of a common sense level to all this work that I think most labs, it's a reason with millions of experiments in molecular biology, there's been no fundamental problems or accidents. In part, the ethics of the scientists doing this, how they train their own scientists, and the simple guidelines that are out there. I think they work very effectively.
So I teach a course on responsible conduct of science, which is based on some of the NIH literature. And in addition, in every other standard course that I teach, I inject some both lectures and exercises where they have to seriously understand it in order to do well in the course. And I would say in addition, iGEM and SynBERC, as part of our training, includes what's called a human practices component, where really they are judged in their competition as to how well they conquer that particular knowledge base as well. So I think there's certainly room for improvement, but there's some hope as well.
I want to say thank you for the question, because I made a comment earlier to someone that I'm here to learn myself. I didn't realize until recently, I don't have NIH funding, therefore my students are not required to have any training in ethics. Because of our affiliation with SynBERC, they have exposure to it in a way that I don't think, or that I think very few other students who are trained as engineers traditionally have. And it's a gap, I think, in terms of how we think about training the next generation of engineers to have a focus on what our responsibilities are beyond the science. And so certainly it's something to think about.
That's terrific. Actually, I think we need to adjourn, but as we adjourn, to reconvene at 1:45, I believe, please join me in thanking Drs. Prather, Church, and Venter.